Lysozymes, also known as muramidase or N-acetylmuramide glycanhydrolase, are glycoside hydrolases. These are enzymes that damage bacterial cell walls by catalyzing hydrolysis of 1,4-beta linkages between N-acetylmuramic acid and N-acetyl-D-glucosamine residues in a peptidoglycan and between N-acetyl-D-glucosamine residues in chitodextrins. Lysozyme is abundant in a number of secretions, such as tears, saliva, human milk, and mucus. It is also present in cytoplasmic granules of the polymorphonuclear neutrophils. Large amounts of lysozyme can be found in egg white. C-type lysozymes are closely related to alpha-lactalbumin in sequence and structure, making them part of the same family. In humans, the lysozyme enzyme is encoded by the LYZ gene. Function: the enzyme functions by attacking peptidoglycans and hydrolyzing the glycosidic bond that connects N-acetylmuramic acid with the fourth carbon atom of N-acetyl-glucosamine. It does this by binding to the peptidoglycan molecule in the binding site within the prominent cleft between its two domains. This causes the substrate molecule to adopt a strained conformation similar to that of the transition state. According to Phillips' mechanism, the lysozyme binds to a hexasaccharide. The lysozyme then distorts the fourth sugar in hexasaccharide into a half-chair conformation. In this stressed state, the glycosidic bond is easily broken. The amino acid side chains glutamic acid 35 and aspartate 52 have been found to be critical to the activity of this enzyme. Glu35 acts as a proton donor to the glycosidic bond, cleaving the CO bond in the substrate, whereas asp52 acts as a nucleophile to generate a glycosyl enzyme intermediate. The glycosyl enzyme intermediate then reacts with a water molecule, to give the product of hydrolysis and leaving the enzyme unchanged. Role in disease: lysozyme is part of the innate immune system. Reduced lysozyme levels have been associated with bronchopulmonary dysplasia in newborns. Children fed infant formula lacking lysozyme in their diet have 3 times the rate of diarrheal disease. Since lysozyme is a natural form of protection from gram-positive pathogens like Bacillus and Streptococcus, a deficiency due to infant formula feeding can lead to increased incidence of disease. Whereas the skin is a protective barrier due to its dryness and acidity, the conjunctiva is, instead, protected by secreted enzymes, mainly lysozyme and defensin. However, when these protective barriers fail, conjunctivitis results. In certain cancers, excessive production of lysozyme by cancer cells can lead to toxic levels of lysozyme in the blood. High lysozyme blood levels can lead to kidney failure and low blood potassium, conditions that may improve or resolve with treatment of the primary malignancy. History: the antibacterial property of hen egg white, due to the lysozyme it contains, was first observed by Lashchenko in 1909, although it was not until 1922 that the name lysozyme was coined, by Alexander Fleming, the discoverer of penicillin. Fleming first observed the antibacterial action of lysozyme when he treated bacterial cultures with nasal mucus from a patient suffering from a head cold. The three-dimensional structure of hen egg white lysozyme was described by David Chilton Phillips in 1965. When he obtained the first 2E-NGSTRA paragraph M resolution model via X-ray crystallography. The structure was publicly presented at a Royal Institution lecture in 1965. Lysozyme was the second protein structure and the first enzyme structure to be solved via X-ray diffraction methods, and the first enzyme to be fully sequenced that contains all 20 common amino acids. As a result of Phillips' elucidation of the structure of lysozyme, it was also the first enzyme to have a detailed, specific mechanism suggested for its method of catalytic action. This work led Phillips to provide an explanation for how enzymes speed up a chemical reaction in terms of its physical structures. The original mechanism proposed by Phillips was more recently revised. Chemical synthesis: the first chemical synthesis of a lysozyme protein was attempted by Professor George W. Kenner and his group at the University of Liverpool in England. This was finally achieved in 2007 by Steve Kent at the University of Chicago, who made synthetic functional lysozyme molecule. See also, egg allergy, references. External links: Muramidase at the U.S. National Library of Medicine. Medical subject headings: Lysozyme, enzyme, sequence, crystallization, structure. Lysozyme.co.uk. Retrieved January 4, 2009. Proteopedia.org. HEW Lysozyme, directory, lysozyme chloride.